Just a quick overview of what the normal timeline for development, just talking about CMC, chemistry, manufacturing, and controls, to get you to phase 1 of the clinic. There are a number of pieces. Analytical methods need to be developed, upstream, downstream, that is the fermentation and purification need to be developed. It has to be formulated so that the material can be given to people. And of course, then it has to be manufactured. And lest we forget, the paperwork has to go in. The IND has to be written, regulatory has to get that accepted, regulatory agencies have to review it and accept it. We're gonna hope I get this right. What's a little bit harder to see, and I apologize, it looks much easier to see on the screen, is historically quality was viewed as coming late to the game. So what you can't see are these much, or more difficult to see, these light squares, and that represents where quality was usually believed to be involved in the whole CMC pathway. So you can see all of it's pushed way down here towards the end. I contend that's not where it belongs, or at least not the only place it belongs. Okay. All right. The role of quality. Certainly one of the big issues very early in quality and the boring part of quality, if you will, is dealing with suppliers. When you're developing, early in development at least, and you're picking suppliers, you need to pick a supplier that will be able to take you through the life cycle, take you to the commercial. Because as we've just seen, timelines are getting much smaller, things have to get to the clinic faster, and once you've made a commitment with a particular vendor, it is very difficult to make those changes on the fly. So, quality has to manage the supplier quality. Some of it's the really boring agreements, all those documents that have to be gotten into shape and signed, who's responsible for what, who's authorized to make what changes. And equally as important, minimum requirements have to be agreed upon. What does the product have to look like? What does the material you receive from the vendor or the work that they're doing, what does it have to look like? Often, industry goes, quality agreements, we do that late. When you get into commercial, we'll do that. We're not going to talk about quality early. That's changing, and it has to change. Recently, the agency, the FDA, has come out with a statement about quality agreements. And yes, they're talking about them being late, but they're also encouraging companies to think about quality much earlier in the process. And if I sound like I'm really into quality, it's because I am, and I apologize. I'm not going to read you the federal regulations and what it says, but basically what the government has said is you need to consider early in the process your supplier agreements, the quality of what your supplier can produce, and in this case, we're not just talking about the quality in terms of what does the protein look like or how pure is it, but really the kind of information that they can bring, their understanding of the kinds of concerns that you have. Your supplier has to be a partner in a way that didn't used to be the case. Additionally, one of the parts of quality early is identifying quality requirements. Not only the assays, but also the specifications. Here's a table that lists a whole slew of kinds of protein analytical methods. Not all of them are appropriate early in the process. And part of what quality should be called upon and should be helping you do is identify which of these methods get you the information you need to have early in the process. Yes, late in the process you're going to need a lot more, but not at the beginning. You're going to have to set specifications for raw materials. Sounds boring, but under the garbage in, garbage out theory, if the raw materials you start with aren't good, then the product you're going to produce in the end is unlikely to be good or is going to require a lot more work on your part to fix it. You have to set specs for in-process. While you're making this, decisions are being made continually. Now, some of this is going to change as you're going through your development program. But nevertheless, you have to set those specs, and quality is part of that. And of course, your release specs. But the really exciting part of quality, and the reason that quality increasingly is important early, is the increased emphasis on quality by design. In other words, quality isn't something that just happens, it's something you put into, engineer into the process and the product from the beginning. Now, I'm going to give a really quick discussion of quality by design because I didn't have as much time as I might have liked. Basically, the whole idea behind quality by design is that you can't test quality in. You have to manufacture something that meets the needs and ultimately the needs of all of your customers. And for a product such as a vaccine, the customer isn't DARPA. Really, the customer in the end is the patient. It has to meet the patient's needs. And sometimes you don't know. And as expressed early, people are sitting there, okay, how often are you going to inject this? How much are you going to inject? Are you going to have to send this into areas where there isn't refrigeration and you have to have something that's stable at high temperature? All of those are concerns and are part of setting quality requirements for the product. You have to set your target profile, your product profile. And those are those kinds of questions we were talking about. Early in discovery, you're going to be identifying a molecular target. That seems sort of silly to say, but in fact that's the case. Is it going to be glycosylated? Isn't it going to be glycosylated? How big is it? How small can it be and still be effective? What does its three-dimensional structure need to be? As you move further down, the target profile is going to get more complicated. You're going to have to be able to specify more information. Your target profile is really what's going to drive the quality by design in cell line development, in any other piece, but I've chosen to focus on cell line development today because we could talk about quality by design for hours. Or at least I could. I suspect most of you would sleep through it. Historically, the quality of the products produced by the cell line was not what was of concern. What was driving it was titer, the amount of protein any particular cell line produced. Now, with cell lines that can produce very high titer, the luxury of being concerned about the quality of the protein has become important. So someone has finally said, well, you know, it's great that we can reduce 8 grams per liter, but 7 of those grams are aggregated and we can't get them to disaggregate. So really, we're producing 1 gram per liter. Or maybe we're producing 1 7 grams per liter, but it's not glycosylated properly, or it's not folded properly, and we can't get it to refold. All of those are now concerns, which are quality concerns, and which have moved much earlier into development. Another area is risk ranking. What is the probability of a particular problem, and how severe would it be if that problem existed? So if you have something that's likely to happen and is going to be a killer to the program, that's obviously a high priority to work on. If, however, it's likely to occur, but its severity is low, that's going to be a much lower risk. You're going to spend less time or energy in fixing that. Additionally, how likely are you going to be able to determine if that particular negative occurrence happens? Can you detect it? Well, if you can detect it, it's much easier. But if you can't detect it, and it's a high severity or a high probability of occurring, then again, that's where your energy goes. So that's another place where quality by design, and in particular quality, has to occur early in the process. The last thing you want to do is develop a process on the bench and get to the point where it is brought to a manufacturer who looks at that and says, that's very funny, now go make something we can in fact produce. We can't detect this error, and this error is likely to occur because of necessity. Applying this quality by design to a clone selection. Multiple clones are created, and they all are producing the protein of interest. But you're going to have to sort between them. You can't take every lead forward. So you have to think about the quantity that they're producing. Yes, the titer does matter somewhat. And I shouldn't say that so cavalierly. Obviously, it does matter quite a bit. But it also has to be functional. The biochemical properties of the material. Is it structured properly? The cell line itself, is it viable? Is it stable? And equally of concern is, can it be produced in a standard medium? Because that has an impact on costs. And when you are thinking about commercial, cost matters. There are a number of common expression systems. There are mammalian, primarily using Chinese hamster ovary cells. But there are E. coli, there are Cereviaceae, there are other microbial systems. One of the decisions is, what is the right system? Right now, a large proportion of them are in varying cell types. Certainly the Pfenex system increasingly, because it is shown that it has some advantages. Productivity is increasing. Literally every year and a half, productivity is about double. That means that that becomes a lower concern, and the quality becomes a higher concern. So just to summarize, the kinds of things that have to be thought about as quality in early development meets the target product profile. The cell line technology, the number of cell lines that can be chosen, the selection of the clone itself, and the media, how long it takes to actually produce a reasonable amount of protein, so the time in which the material must be cultured. The fermentation considerations, the temperature, the DO, pH, the harvest criteria. How do you get the protein out of your cell culture? All of that affects target product issues. The homogeneity of the monomer. Whether or not you're forming multimers, and whether those multimers can in fact be dissociated. The productivity, the expression level. Whether it does the same thing over and over again. Certainly we are hearing that is a consideration for the work being done at MGH. Its performance and the genetic stability of material. So I would contend that there is actually a new place where quality impacts early development. And as you can see, there are a lot more light boxes, and they reach much further forward into development, reaching all the way back into developing the cell line and selecting it. Thank you. I can take questions, or I can wait for the panelists.
Sheila, what's the, in protein production, you're talking about, where have you seen things, the most difficult steps now, where have you seen the most problems?
Interestingly enough, it's almost as though you're chasing things out. Certainly it used to be that titer was the biggest problem. Now increasingly it's actually at the harvest. People are finding themselves with so much protein because the systems are so improved and the media systems are now matched in a much better way that they are not able to actually get all the protein out. The other area, and this is one that is increasing because it's an increasing concern on the part of the regulators, is the higher order structure of protein. Much, much more information is being asked of individuals who are working on proteins in terms of their structure. So the analytical methods are now suddenly becoming I wouldn't say a bottleneck, but certainly an area of concern much earlier in development. It used to be that an SDS page was a suitable method for looking at a protein early in development. That's no longer acceptable.
So what's the newest, hottest technology for looking at protein structure?
Unfortunately, there isn't one. As with protein structure along, it has been multiples. So CD is becoming much more important. DSC is becoming much more important. Hydrogen deuterium and mass spec are becoming important. So you're doing hydrogen deuterium interchange and then looking to see what the exposed sites are.
Do you see the release testing or characterization?
Characterization primarily, but you're being asked for characterization data much, much earlier in the process. Even at a phase 1 submission, questions about the structure of the molecule in a much more detailed way. Yes?
In each batch, let's say the phase 1 product, how much should you save for QC, characterization, and just retention samples?
How much do you lose?
Now, how much should you keep, put aside, save?
All right, well, I'm going to assume that you have already made your laboratory reference. But nevertheless, from your first batch that is at research scale or phase 1 scale, might be the best way to describe it,
Yeah.
you should be making another reference. That actually takes a large proportion of material, several, tens of milligrams.
retain samples and support stability studies, have got to be another thing to factor in.
Stability studies, you know, some of it will depend on the way you're actually going to bottle it in the end. So while I would love to be able to give you an absolute number, I can't. But you're going to have to retain at least enough to do stability for several years. You're going to need to retain, and this is one of the areas that people often skip early, is maintaining stability samples in the drug substance before the drug product. Everybody used to say, well, we don't need to do drug substance early, we're going to go right to drug product. But increasingly regulators are saying, no, 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 no. We want stability on the drug substance as well as the drug product. So you're going to lose material at that point as well. You need to take material for characterization, and depending on the methodologies you use, that's going to be several milligrams. So you could easily lose 100 or more milligrams just for testing. And if you're looking at say, an antibody product where you're bottling 50 mg per mil solution and you're bottling a mil per vial, you want to have 3 years of stability. You have to do sterility on that 3 times. That's 20 vials just for that, plus all of your testing. You can see very quickly that you're losing a large amount of material.